We are going to start our today's lecture, and as said earlier, we are going to have an OSPE. Today, we are going to discuss culture media and biochemical reactions related to bacteriology and microbiology. Uh, Culture media and biochemical reaction, they are two important unobserved stations. So starting with the first spot, I'm just like the previous OSPE, we have bacteriology and microbiology. Bacteriology we have certain colored colonies. Only one mycobacterium is not cultured on artificial culture media, and that is Mycobacterium leprae, that cause leprosy. So, Mycobacterium leprae is one mycobacteria which is not cultured on artificial culture media. It is cultured on live tissues. So, it is inoculated in animals like laboratory rats or mice, and in the footpad of armadillo. ये बुक के अंदर लिखा हुआ है armadillo. So it was that for the first time the armadillo was used for culturing this, but nowadays we can culture this on artificially in the lab, uh, on in mice or rats, and on artificial tissue culture also. So give its use, it is media for mycobacterial species. Identified the colonies. These are off-white, puff-colored colonies of Mycobacterium tuberculosis. Why is the color of this medium light green? Because it contains malachite green. Malachite green is added in this medium to stop the growth of all other gram positive and negative bacteria. Name another medium used for the same bacteria. Same bacteria in this case means Mycobacterium species. So another medium is Middlebrook medium. Middle, Brooke, Brook medium. So usually in our lab and mostly commonly in all the labs, we use LJ medium, that is Levenstein Jensen medium, but in book it is also written that another medium can be used, that is uh, Middlebrook medium. So another spot is this is a culture medium, pink in color. This is McConkey's agar. So this is one of the aggars, and this agar is used to differentiate between lactose fermenters and non-lactose fermenters. Lactose fermenters and non-lactose fermenters are mostly gram-negative rods. So this is a solid culture medium. Solid culture medium is that it contains all the basic nutrients for the growth of the bacteria, like proteins, carbohydrates, electrolytes, salts, and an agar to form this jelly like medium. And in this case, if we have to differentiate between the two bacteria, like lactose fermenters or non lactose fermenter, we have to add an indicator that will indicate whether the lactose is getting fermented or not. So we are adding lactose as an additional medium in the nutrient agar, or the basic along with the basic ingredients, and we are also adding an indicator in case of a culture medium is a substance which is colorless at pH 7, and it is going to change its color, uh, a different color in acidic and basic environment. So whenever lactose is fermented, uh, the environment becomes acidic and the color will change. So the indicator used in this medium is neutral red, which is going to be pink in acidic environment and yellow in basic environment. So if I say that this is a McConkey's agar with a growth of bacteria which are pink in color, and this colony is a lactose fermenting bacteria. The other question may be, okay, what are the lactose fermenting bacteria? The two important lactose fermenting bacteria are E. coli and Klebsiella. So name the medium, McConkey's agar, give its use. It is used to differentiate between lactose fermenters and non-lactose fermenters. Name the indicator used in this medium. It is neutral red. Identify the colony. You just have to write that it is a lactose fermenter bacteria. You don't have to identify whether it is E. coli or Klebsiella, just that it is a lactose fermenter. If it is a yellow colony on the same medium, it is going to be a non-lactose fermenter. Non-lactose fermenter are going to be Pseudomonas, Proteus, and Shigella, Salmonella, and many other. So you have to know the important common disease causing bacteria. Name some important components of this medium. As I already told you, agar, water, electrolytes, uh, sodium chloride, then lactose, uh, proteins in form of peptones, and then an indicator, which is neutral red. This is another medium. This is a basic agar. This is called nutrient agar. Nutrient agar is that this agar contains all the basic nutrients required for the growth of the bacteria. Any bacteria can grow on this agar. This agar is of white in color. All the colonies of any bacteria are going to be of white in color. This agar is going to show you growth of all bacteria, all gram positive and negative bacteria, but you cannot differentiate between the bacteria regarding their specific characteristics. For example, hemolysis or lactose fermentation or any other character. So this is a nutrient agar. This is the basic agar in which if we add certain other substances, we can make other agars. For example, if we add lactose and an indicator, we can make a nutrient, a McConkey's agar, like in the previous slide. Identify the test. So why this nutrient agar is used if we cannot differentiate the bacteria? First we have to differentiate the bacteria on different other agars. Then once the bacteria is grown on the other agar, and we have identified the bacteria as an individual bacteria, we are going to put that bacteria on the nutrient agar, and we are going to put the antibiotic sensitivity. So this picture shows the nutrient agar, and this picture shows discs of antibiotics. So we are going to put discs of antibiotic. The next day we are going to come and see if the disc is having a halo around it. It means that this antibiotic, this bacteria is sensitive to this antibiotic. This is the growth of bacteria, the soft white, and this clear zone means that bacteria is not growing around this antibiotic. If the bacteria is growing around the antibiotic disc, that means that the bacteria is resistant to that antibiotic. So we are going to put a few antibiotics for a certain bacteria, and then we are going to check to which antibiotics that bacteria is sensitive or resistant, and we are going to put that in our report. That is called culture and sensitivity. So we are culturing the bacteria from the specimen and identifying the individual bacteria, and then putting the sensitivity, antibiotic sensitivity, so that the clinician can give a specific uh, antibiotic for that disease. Identify the test. This is an antibiotic sensitivity test. Agar is a nutrient agar. Name the type of the test. It is antibiotic sensitivity test. This is how the question can be asked in a different way. What is NCCLS in your book? This is an abbreviation written, that is National Committee for Control of Laboratory Standards. So this is an international universal committee which gives all the SOPs and protocols or criterias for biological testings. So NCCLS means that National Committee for the Control of Laboratory Standards. So the compositions of the agar, the types of the agar, the antibiotics used for certain bacteria, and the halo around the disc, all the standards are used, all the standards used around the world are from this NCCLS. What is McFarland standard? This is that when we are putting the antibiotic sensitivity test, we can put bacteria and spread it on this agar. This is called carpet technique. Carpet technique, it is that we are going to take a colony from another agar. For example, uh, there is a sample of urine, and we have put that sample on a culture media. The next day, if we see a growth, and we have identified the bacteria, we are going to put the sensitivity for that bacteria. We are going to take a colony from that uh, agar. We are going to put it on this nutrient agar. We are going to put these discs and then incubate it again for 24 hours and then see the result. This technique is called carpet technique. Another technique is that if we take the colony and instead of spreading it on this agar, we are going to put it in a peptone water, dissolve that colony in that peptone water, and then drop or spread that peptone water on this culture, on this nutrient agar plate. That is going to spread again on the whole plate like a uniform uh, substance. So in that case, McFarland standard is that how much colony you are taking from a specific agar to put on a nutrient agar. Because if you take a colony uh, which is larger in amount, that may form growth around a disk, and that may give a false positive or a false negative result. So a standard is that we are going to dissolve barium chloride in water or a distilled water, and we are going to check our colony or the haziness of the colony against that barium chloride solution, which is taken as a standard. If our colony is more hazier than that barium chloride solution, that means that we have put a larger amount of colony. And if the haziness is less than the barium chloride solution, that means that we have put a less amount of colony. So why standard? Standard is maintained because the antibiotic can be given in a proper dose, and a specific antibiotic can be given at any center around the world for a particular bacteria. So McFarland standard is actually a barium chloride solution which is taken as a standard. You don't have to go into the details of the amount of barium chloride. Just knowing that what is McFarling standard, it is that we make a solution of barium chloride, and we compare the haziness of that with our haziness of our colony which we are putting on the nutrient agar. This is another culture plate. This medium is blood agar. Again, it is a nutrient agar, but the only added thing is blood. So it will form a blood agar. What is the use? It is used to differentiate between hemolytic and non-hemolytic bacteria. So if the bacteria is hemolytic, it can be beta hemolytic bacteria or an alpha hemolytic bacteria. Gamma hemolysis is actually a misnomer because gamma hemolysis means no hemolysis. So if it is no hemolysis, so gamma hemolytic organism cannot exist. So name a gamma hemolytic organism. This is a common thing that we speak and we ask, but this is actually not a proper question. The question should be, I have written intentionally because the question should be that name non-hemolytic organisms on blood agar. So the hemolysis or hemolytic organisms can only be alpha or beta hemolytic. So in cases of alpha hemolysis, there is a clear zone of hemolysis around the colony. So in case of A, if you see, there is a clear zone around the colony. But in case of B, there is a green tinge because not all the RBCs, they are hemolyzed. This is called alpha hemolysis, and alpha hemolysis is because of the production of peroxidases. The beta hemolysis is because of the production of hemolysin by the bacteria. So in on this agar we can differentiate hemolytic and non-hemolytic, and in cases of hemolytic we can differentiate whether it is a beta hemolytic bacteria or a alpha hemolytic bacteria. The other questions can be name some beta hemolytic or alpha hemolytic bacteria. So read the book and make a small table for bacteria which are positive for alpha hemolysis and beta hemolysis, and whatever I'm going to tell you in the further slides. So make your own list of bacteria so you can remember those. So you have to make a list for beta hemolysis, alpha hemolysis, lactose fermenters, non-lactose fermenters till now. So again, whenever making a list for yourself, the most important thing is that you have to know the common bacteria. No one is interested in a long list of 10 bacteria. Even if you speak or remember two or three common bacteria, that is going to give you full numbers, and that is enough, because you are going to see all those common diseases in the ward. What is the indicator used in this medium? There is no special indicator added, but RBCs themselves are the indicator, because RBCs are going to show hemolysis or no hemolysis. So RBCs themselves are the indicator. This is a biochemical reaction or a biochemical test. This is in the form of an agar. Again, this is a nutrient agar. Only thing added in this is a citrate and an indicator. Citrate is that this test is called a citrate test, or this agar is called citrate agar. This agar is green in color. This is having all the basic nutrients along with citrate 1 and an indicator which is called bromothymol blue. Bacteria which utilize citrate, they are going to make the environment alkaline. Bromothymol blue indicator is going to turn blue. So if citrate is consumed, environment is alkaline, the agar is going to turn blue. So if we put a colony or the sample on this agar, this is the original color. If the citrate is not consumed, it is going to stay green. The test is negative. If it turns blue, the test is positive. Indicator is bromothymol blue and citrate is used. If we ask another question, that name citrate positive bacteria, that is the next list you are going to make. The most common ones are Klebsiella and Vibrio cholera. Which indicator is used in this medium? That is bromothymol blue. Name another medium or agar with the same indicator, that is clad agar or clad culture media. Clad is a culture media used for culturing the urine sample. I'm going to show you in the next slides. This is another biochemical test. This test is again in the form of an agar or a solid medium. Name the biochemical test. This is urease test. Again, this is in the form of an agar. The agar is containing all the basic nutrients. The added thing is urea. We are differentiating which bacteria consume urea and break down that urea because of production of urease. So actually we are differentiating uh, urease positive bacteria from those which are not producing urease. Two most important bacteria producing urease enzyme. Number one is H. pylori. So Helicobacter pylori, which cause diseases in the stomach, and four important diseases caused in the stomach by H. pylori are gastritis, acid peptic disease or peptic ulcer, adenocarcinoma of the stomach, and malt lymphoma of the stomach. So these are the four diseases related to H. pylori, and two tumors which are related to a bacterial etiology. And the pathogenesis of H. pylori is related to the production of urease. So you have to name that bacteria first. Proteus is another bacteria which produce urease enzyme. So make a list of two or three bacteria which produce urease. So the normal color of urease agar is off white. If you put a colony and there is no breakdown of urea, it is going to stay off white. But if there's production of urease and breakdown of urea, the environment is going to turn alkaline and the agar is going to turn pink. So that means that there must be an indicator in this agar, and that indicator is phenol red. Phenol red is going to turn yellow in acidic environment and pink in basic environment. So identify the test, urease test, give interpretation of the tube on the left hand side, that is positive. Give names of the organisms which are positive, which indicator is used in this medium, that is phenol red. Name another biochemical test with the same indicator, that is TSI, triple sugar iron test, that we are going to discuss in another slide. So this is a nutrient agar. Give its use, antibiotic sensitivity test. Name the indicator used in this medium. There is no indicator used in a nutrient agar. Give the composition of this medium. You have uh, the basic, all the basic ingredients: water, agar, calcium, magnesium, sodium chloride, peptone acting as proteins, and certain carbohydrates. Nutrient agar is used for antibiotic sensitivity testing. Name some anaerobic bacteria. Most common are all the Clostridium species. So all the Clostridium species, they are anaerobic. So names, make a list of some other anaerobic bacteria also, and emphasizing here that anaerobic means lack of oxygen, aerobic means presence of oxygen. Name this medium. This is chocolate agar. This is nutrient agar with blood, and if we heat that blood agar, it is going to turn brown because of the production of hematin. So we are going to heat it at 60 degrees centigrade. We are not going to boil it because we don't want hemolysis. We only want hematin. Give its use. It is used for certain samples. It is used for culturing bacteria which need hematin. It is used for culturing bacteria which are capnic. Capnic are carbon dioxide loving bacteria. And list the composition of this agar. This is a nutrient agar with blood and with heating. Chocolate, the name chocolate is because of the color of this agar. So what is the indicator used in this medium? There is no indicator used in this medium because the colonies we are identifying have their own specific characteristics. Name three capnic and capsulated bacteria. Capnic, carbon dioxide loving, and capsulated, they have a capsule around them. Three important bacteria, they are Streptococcus pneumoniae, Haemophilus influenza, and Neisseria species, both Neisseria meningitidis and Neisseria gonorrhoeae. So these are capnic, carbon dioxide loving, these are encapsulated, and these are cultured on this chocolate agar. Uh, the carbon dioxide rich environment is produced by putting the culture plates in a candle jar. Candle jar is just a tin jar. You are going to put the culture media, you are going to lit a candle, and the candle is going to consume the oxygen. You're going to put a lid on that. The candle is going to consume the oxygen and produce carbon dioxide. That is the enough carbon dioxide which is required by these carbon dioxide loving bacteria. We are not saying that there is no oxygen. We are not saying that the environment is a vacuum. That is only that carbon dioxide is more than the normal uh, environmental carbon. These are important bacteria, and they cause infection in general tract, in meninges, in the respiratory tract. So the samples coming from these areas are cultured on this chocolate agar along with blood and McConkey also. So CSF swabs from nose, throat, ear, and swabs from vagina and cervix in cases of Neisseria can be cultured on the agar. Please make one thing very clear, that we are not putting bacteria on the culture plate. We are putting the sample on the culture plate, and we are actually putting the sample to identify the bacteria. When we put the sample, we are going to come the next day after 24 hours, and then we are going to see which bacteria is on the plate, and we are going to identify it through biochemical test and staining. Name the test. This is indole uh, test. Interpret the test in tube A and B. This is a liquid medium. This is peptone water. And indole, the name of the test is indole test. In this case, there is formation of ring in both negative and positive. So make it very clear that there is ring formation in both the cases. But if the ring is red, it is a positive test. If the ring is yellow, it is a negative test. Now what is the principle of the test? This is that you are going to put tyrosine amino acid in this peptone water. Bacteria consuming that and breaking down tyrosine is going to produce some byproducts which are laid on the surface of this liquid. The next day, after incubation, you are going to put a reagent which is called Kovacs reagent, KOVASC, Kovacs reagent. That Kovacs reagent is going to react with the byproducts produced after the breakdown of tyrosine. If the tyrosine has been broken down, byproducts produced, Kovacs reagent is going to produce a red ring on the surface. If not, the Kovacs reagent is going to float on the liquid as a yellow ring. So red is positive, and this yellow is negative. Name the organism giving a positive result, that is E. coli. To differentiate between E. coli and Klebsiella, in case both are lactose fermenters, we can perform an indole test. Name some catalase positive bacteria. Name some oxidase positive bacteria. Name some coagulase positive bacteria. So please make a list from the book for your own self regarding all these questions. And we are going to discuss in the lab, and you can come up with any questions if you have. So before going to this last slide, one of the culture medias left is clad agar. Clad agar is light green in color. Clad means cysteine, lactose, electrolyte deficient media. Clad is basic nutrient agar containing lactose and cysteine, but there are no electrolytes. This is a specific media just for urine samples. So if we have urine samples, we are going to put that on clad media. Clad media is used to differentiate between lactose fermenters and non-lactose fermenters. It is same as McConkey's agar, that we are differentiating between lactose fermenter and non-lactose fermenters. But in case of clad, only the indicator is different. The indicator used is bromothymol blue. The agar is light green in color originally, and if the lactose is fermented, the colony is going to turn yellow. If the lactose is not fermented, the colony is going to turn blue. Yellow means that there is acidic environment, and blue means that there is basic or alkaline environment. Agar is clad. Use is for putting culture of urine sample. Why? Because to differentiate lactose fermenters from non-lactose fermenters. Lactose fermenters remains the same as on McConkey's agars, that are E. coli and Klebsiella lactose fermenters, and Pseudomonas proteus, Shigella salmonella, they are non-lactose fermenters. This is a specific media just for the urine samples, and we are detecting lactose fermenters and non-lactose fermenters because urinary tract infections are mostly caused by gram-negative rods, and 90% of the UTIs, they are caused by E. coli. Now, coming to this last slide and last test, this is an important test to differentiate various gram negative rods. Name the test. The test is TSI, triple sugar iron test. TSI test. Name the indicators used in this biochemical test. So, the indicator used in this test is phenol red. So phenol red is going to turn yellow in acidic environment. It is going to turn red in basic environment. Test is TSI, triple sugar iron. Indicator is phenol red. Triple sugar means that there are three sugars: glucose, lactose, and sucrose. Sometimes it is written in the book lactose, glucose, and maltose, or mannose. But commonly it is glucose, lactose, and sucrose. If it is maltose, even then glucose and lactose are the basic sugars which are present. So there are three sugars. There is an indicator, and it is in the form of a solid agar, and it is put in a tube like this shown in the picture. Which sugars are used in this test, I've already told you. And now coming to the interpretation, it is a solid agar with basic nutrients, with an indicator, and with three sugars. We are actually differentiating gram negative rods in this test. There are four results. That is, if we put a colony from a culture media to identify the bacteria, we are going to put a colony in this agar. If you see that this agar is put in this tube in such a way that there is a solid portion on the base, and then there is a slanting portion above that. This solid portion on the base is called butt, butt, and this slanting portion is called a slant. So in cases of citrate and urease test in the previous slides, the agar was put only in slant, but in this case it is put in this two forms. In the bottom one is butt, and then there is a slanting portion. We are going to put a colony. We are going to incubate it at 37 degrees centigrade in the incubator. The next day when we are going to come, we can have four different result. Number one is that there is no fermentation of any of the sugars. The environment is going to stay alkaline. So the phenol red is going to turn pink. So number one result is that it is going to turn pink. Both butt and slant are going to turn pink, indicating that neither of the three sugars have been fermented. Example is Pseudomonas. So Pseudomonas is one bacteria which is fermenting no sugar present in this test.
Sorry to
Then we
interrupt
have,
you. Just 4 minutes left.
okay, the next is that if one or two sugars are fermented, and that one means that glucose is always fermented first. In case of two sugars, the next sugar is going to be lactose. So if glucose, if one or two sugars are fermented, or even if only one sugar is fermented, first of all, there is going to be an acidic environment because of the because of the fermentation of a sugar. Phenol red is going to turn yellow, but because the byproducts are not enough to maintain that acidic environment, the slant is going to turn pink because of the presence of oxygen. So, in this case, in our second interpretation, the butt is yellow, but the slant is pink because there is acidic environment because of the fermentation of one sugar in the butt, but there is oxidation of the byproducts because of the presence of oxygen which cannot penetrate the butt on the slant. So the slant is going to turn pink. So two bacteria which are going to ferment one sugar, glucose, that are Shigella, Salmonella, and Proteus. So in case of Shigella and Salmonella, uh, this the tube is going to show yellow butt and pink slant. In case of salmonella, there is going to be a blackening in on the slant due to the production of H2S gas. But if there is a lot of blackening, this is Proteus. So you cannot identify the reaction in the yellow reaction in the butt in case of Proteus. So if it is pink slant, pink butt, pseudomonas. If it is yellow butt, pink slant, it is shigella or salmonella. If there is blackening on the slant, it is salmonella. If there is a massive blackening with a small portion of pink slant, it is Proteus. The last reaction is that the entire medium is yellow in color. That is when lactose, two sugars, or all the three sugars are fermented. And in this case, the acidic environment is massive, and it is not going to turn pink even in the presence of oxygen. So the byproducts are going to cause the phenol red to turn yellow because of the excessive acidic environment. In this case, the two bacteria are E. coli and Klebsiella. And to differentiate these two from each other, the identification point is that if you have a yellow butt, yellow slant without gas production, that is Klebsiella, and if you have a gas production which is carbon dioxide gas, it is E. coli. The carbon dioxide gas is going to produce bubbles in the agar, or it can just cause the agar to scratch away from the sides of the tube. So this is TSI, triple sugar iron. You have an indicator, you have four results. So you can have any of these one tubes in exam in case of Ospe, and you are going to, you may be asked these certain questions. You may have this picture having all these four tubes, and if there is numbering 1 2 3 4 or A B C D, you may be asked that give results of all the four or result of any of one of them with examples.
Sorry, sir.
So number
Lecture
one
time is
is,
over now.
okay ji, Allah Hafiz.